In this video we will discuss about the different combinations that we have discussed when we are discussing the figures of related to this paper. In this figure what they did is they combined all those successful approaches and try to find out whether they will have a synergetic effect on this allogenic NK cells, not in terms of suppressing the immunogenic response of the host NK cells and the T cells, but at the same time also maintaining this allogenic NK cells cytotoxicity against the cancer cells. So the different approaches that they have combined are this HLA knockdown on this allogenic NK cells. HLA is this human leukocytes antigens, and this SCE, this is the customized peptides with mutations, and at the same time it also has this HLA-E part that is overexpressed, and this PD-L1 overexpressions. And so the panel A shows the results about the co-culturing of these allogenic NK cells with the different combinations of this HLA knockdown, SCE overexpressions and PD-L1 overexpressions with this CD8 plus T cells or primed CD8 plus T cells with cytomycins treated NK cells for 7 days and in the presence of IL-2. So what the result shows is that for the control, where they use the untransduced allogenic NK cells, they see the maximum cell lysis, and with the knockdown of this HLA knockdown using this shRNA1 and shRNA3, they see a minimum cell lysis. However, the cell lysis increased when they overexpressed SCE or PD-L1, but the increased is higher when the SCE is overexpressed. It can be seen for shRNA1 and shRNA13. And so this results indicates that the immunogenic effect from CD8 plus T cells can be suppressed just by suppressing HLA knockdown. And when they check the results of co-culturing these allogenic NK cells with the donors or host NK cells, and they are able to see that in the case of control, where they don't use any transductions, cell lysis is minimum, and those cell lysis is remain minimum or remain at the lower rate when they overexpressed when they suppress this HLA knockdown. And however, this slightly increased for the effector to target ratio is 10:1, but they are mainly remains at the lower range. But these things they are mainly remain at the lower range even with the overexpressions of this SCE that can be seen here. And so here the only results for the HLA knockdown is not shown. The results with the combination of either SCE or PD-L1 is shown. So when the PD-L1 is overexpressed, they see a more cell lysis, indicating that this PD-L1 overexpressions cause more cell lysis or immunogenic effect by the host T cells against the allogenic host NK cells against the allogenic NK cells. So the best combinations right now seems to be the HLA knockdown with SCE overexpressions. And now they wanted to check at these conditions whether this allogenic NK cells has enough cytotoxicity against the tumor cells or not. So they check this allogenic NK cells co-culture with the K562 cells, and then they are able to see that in the case then they are able to see that either the HLA knockdown, HLA is overexpressed or knocked down in the allogenic NK cells. The cell cytotoxicity against these K562 cells are minimum, but when they are combined with either SCE or PD-1 overexpressions, the cell cytotoxicity or the allogenic or the NK cells cytotoxicity against the K562 cells increases. So it is essential to have this overexpression of SCE or PD-1 to maintain the cell NK cells cytotoxicity against the cancer cells. So this is what the final conclusions that it comes from. The results for this allogenic NK cells with the cancer K562 cell lines is shown in the supplemental figures. You can refer to the supplemental figures from the descriptions that has been provided from the descriptions of the paper that has been provided in the link of the paper that has been provided in the descriptions. So that's all for this video. Thank you for your time.